new tissue-engineered airway. Just like that. So we've been able to demonstrate then that it's possible to produce a functional organ by combining a donated decellularized scaffold with a patient's own cells. So not only was this the first example of a transplanted windpipe, it was also the first example of a tissue-engineered organ made from a patient's own stem cells. So it's hoped in the future that more patients can be treated with engineered trachea in a similar kind of way, and also that a lot of the principles that were developed here can be applied to regenerate other organs. So for example the larynx, which is simply further up the airway, and also other hollow organs such as the colon, the bladder, blood vessels maybe in the future. So I'd just like to mention all of the different people that were involved in this interdisciplinary team. They all had special skills which they brought to the project, and without all of them it wouldn't have been possible. So Professor Paolo Macchiarini was the main leader and driver behind this whole project, and was also the surgeon responsible for the implantation. The team in Milan, they developed the bioreactor, and the team in Padua were responsible for perfecting and carrying out the decellularization of the donor trachea. And then the two teams in Bristol, we were involved in the cell biology, so producing the chondrocytes and the epithelial cells. And perhaps the most important person in all of this was definitely the patient herself, who had the courage to go ahead with this operation, and hopefully she's now benefiting from her bravery. So I'll just finish by acknowledging those people again, and also the other members of the Bristol Stem Cell Biology Group, and also our funding bodies. Thank you.
Thank you for a fantastic talk. So we have, before coffee, we have time for 2 or 3 questions.
Well, congratulations again. It's a fantastic publicity for tissue engineering. Do you really think that you can produce any organ with this technology? The reason I'm asking, and there is a question to the second half of the sentence, is that I'm really surprised that for 2 weeks, although you showed 2 weeks after the operation, blood vessels appear. But 2 weeks, if there is no blood supply, if there are no nutrients delivered to those especially to the epithelial cells, they would die. So what happened? And there are other organs where this simply will not work because you cannot wait for such a long time.
I don't think we actually waited that long. That was just the time that the bronchoscopy was taken that I showed. It did happen much quicker than that, but we didn't have the footage to show of that. So no, it did happen much faster than that.
So you think this is a panacea for every organ?
I don't know about that, but I think certainly for hollow organs, I think a lot of the principles that were developed here should be able to be applied for generating hollow organs. Obviously solid organs are a lot more complex, which is why we might need some blood vessels involved as well. Yeah.
Thank you.
Hi, how much did the surgery cost, and do you think it's actually feasible to be done for everyone who's experiencing this type of problem?
Yeah, it was quite expensive, and we didn't get any funding for this at all. It was basically each of the groups just provided their time and expertise, basically because it was a one-off patient. And at the moment, with the methodology, then no, I don't think it really is feasible to carry out it on a routine basis. The whole thing needs to become much quicker and a lot more automated. So maybe some of the bioreactors that we saw in the first talk in this session would be very useful in something like that. The whole process does need a lot more automation.
Did you ever get any money back from EasyJet when you had to fly out?
No, we didn't actually. I think they refunded the cost of the ticket, which was like £15 or something. But no, so the Vice Chancellor of Bristol University actually paid for the jet in the end. We had to hire a private jet to take it over to Barcelona.
Thank you. So we can continue this conversation during the coffee break.